welcome everyone. Thank you so much for coming to Grand Rounds today. A couple announcements before I introduce our speaker today, Dr. Danielle Bassett. So first, our speaker next week will be our very own Dr. John Mann, who will be presenting on how to flatten the curve of suicide deaths in the US, lessons from COVID-19 missteps. And now I'm going to turn it over to Barbara Eddington for a quick announcement, and then I'll introduce our speaker.
Great, thank you so much. I appreciate you inviting me. We are in that season again. It is time for you to be nominating yourself for the US News survey. We have not yet gotten the actual start time for the voting. We understand they have sent out some paper ballots, so some of you may have received those, and they may be rolling out the online voting in February, maybe starting February 12, 13, 14, we're not really sure, but we obviously will let you know as soon as we know. And I'm speaking on behalf of the New York Presbyterian Marketing Department. Oops, I'm sorry. The New York Presbyterian is now ranked 4th in the nation, which came up from number 10 about 3 years ago. The hospital rankings are based on specialty rankings, which is the reputation score. And as you know, for psychiatry, 100% of your score is based on reputation. So that's the number of times you are nominated by other experts in your field across the country. We have learned that less than 15% of eligible voters actually vote in Doximity. What I saw on the US News website was that they estimate about 26,000 voters are in the Doximity database in your specialty. And they say that of our eligible voters, we have 86% registered. So let's pretend that if only 15% of the total Doximity voters voted, that would be 3,900 votes. And we have 584 possible of that. So we could actually account for 15% of all votes. We are now ranked number 4 in the nation. And according to the US News, they're saying that an average of at least 5% of the specialists need to mention us. And of course, we could be representing 15% of that. We also are holding at a very excellent score in the expert opinion. So at this point, since the voting hasn't opened, I'm here to ask you to give it a test run. For those of you who don't use Doximity, except this once a year, you probably need to refresh your memory of what your password is. Try to log in. If you can't get in, don't remember your password, try to reset it now. The other thing we have seen in past years is that if your profile is not up to date, and specifically if your current license is not in your profile, you will be considered out of date and not eligible to vote. So if you have any issues logging in now or having your profile up to date, let us know. We can help you. We will help troubleshoot with Doximity. Once they start rolling it out, you'll get an email from Doximity. Just click through that, look for the little bell that says you have a notification. And once you get into the notifications, you'll find the Best Hospitals icon. This is where you can vote. You can nominate up to 5 hospitals. When you're looking for New York Presbyterian, you can either just type it all in and it'll fill for you, or you can search for Columbia and Cornell, or just Cornell. But unfortunately, if you just search for Columbia, you'll go to Columbia, Missouri. So that's just a little primer on how to vote. And again, if you have any trouble getting into Doximity now, let us know and we'll work with you. We are available at vote@nyp.org. Thank you.
Wonderful. Thank you so much, Barbara. You are 100% right that it makes sense to try out one's Doximity account. I think I spend about half of my workday trying to figure out passwords, so I'm sure that I'll that one.
Great. Thank you.
Okay. It's a real pleasure to introduce today's speaker, Professor Danielle Bassett. Dr. Bassett is the J. Peter Skirkanich Professor at the University of Pennsylvania. She has appointments in the departments of bioengineering, electrical and systems engineering, physics and astronomy, neurology, and psychiatry. Dr. Bassett is best known for her work blending neural and systems engineering to identify fundamental mechanisms of cognition and disease in human brain networks. Dr. Bassett is also currently working on a book for MIT Press entitled Curious Minds. She received her bachelor's degree in physics from Penn State University and her PhD in physics from Cambridge University, where she was a Churchill Scholar and a NIH Health Scholar. Dr. Bassett has received numerous prestigious awards. I can't name all of them or I would be here all day, but a few are, she was awarded the APA's Rising Star Award, the Alfred P. Sloan Research Fellows Award, the MacArthur Genius Fellowship, and the National Science Foundation Career Award, along with many, many other awards. Dr. Bassett is the author of over 300 peer-reviewed publications and has garnered over 26,000 citations. Although Dr. Bassett's work is probably best recognized in the areas of neural systems, she's also conducted pioneering work on gender misbalancing and citations of scientific articles. And in talking with her just prior to Grand Rounds, she pointed out to me that much of that work was spearheaded by Jordan Dorkin, who is now a professor in our department working in Melanie Wall's division. So with that, we're very excited to hear from you, Dr. Bassett, and I will turn it over to you.
Wonderful. Thank you so much for the very kind introduction. And let's see if I can share my screen here. Great. Okay, so the work that I wanted to share with you today is framed in the context of current advances in computational psychiatry. So as you just heard, my background is in physics. So I come to the brain from a physics perspective and a computational perspective. And so computational psychiatry is a space in which I feel particularly excited and inspired. What I want to focus on within that particular context is understanding cognitive control as a network process. And I'll try to walk you through why I think it is a network process and what I even mean by the term network process. But before that, what I wanted to say is that why cognitive control? Well, cognitive control is interesting to me because deficits in cognitive control specifically, but also as executive function more broadly, accompany many psychiatric conditions. And the fact that it's impacted transdiagnostically makes it a particularly interesting cognitive function to be studying. Just to define our terms, I'm using the phrase cognitive control to indicate the process by which goals or plans influence behavior. And I'm using the term executive functions as a set of cognitive processes that are necessary for the cognitive control of behavior, selecting and successfully monitoring behaviors that facilitate the attainment of chosen goals. Examples would include task switching, working memory, decision-making, and inhibiting inappropriate behavior. Now, when I think about cognitive control, many of these functions seem to be a little bit disparate. And so I want to boil it down to what the very precise process is that's required for cognitive control. And I think something that is shared across many of those examples that I just gave you is that cognitive control requires carefully controlled state transitions. And those state transitions are transitions in cognition, but also transitions in neural states. So if we just think about decision-making, for example, the first state might be weighing options, criteria, and values, and then the second state is deciding on a course of action. That transition is sort of the essence of decision-making. And in task switching, obviously, we start with task 1, that's the first state, and we switch to task 2, that's the second state. Similarly, if we think about inhibiting inappropriate behavior, the first state is the urge to engage in a particular behavior, and the second state is inhibiting that inappropriate behavior. So the fact that each of these has sort of a two-state, at least a two-state process, makes me ask the question of how do state transitions really happen? And that's what I want to focus the majority of my talk on today. And I'm going to break down my comments into three different sections. In the first section, I want to develop a theory for state transitions and their control, and I'll be highlighting some work really in theoretical physics. In the second section of the talk, I want to use that theory to probe normative development versus the psychosis spectrum. And then in the third section, what I'd like to do is to use that theory to probe executive function and dysfunction. And I'll be mentioning some work in schizophrenia at that last section. So to frame this first part, I wanted to read for you a passage from Aristotle's Metaphysics, book 12, sorry, where he says, "Mind thinks itself because it shares the nature of the object of thought. For it becomes an object of thought in coming into contact with and thinking its objects, so that mind and object of thought are the same." And I think that motivates the interesting question of what are brain states? How do they become? So both mind becomes matter and is matter, but how does the mind actually become something? Mind becomes something as the brain represents objects, events, experiences, or concepts. And when I think about these representations or these states, I think of them as a vector of activation, let's say across neurons or across neuronal ensembles. And in each neuron or each neural ensemble, there's a particular level of activity, which I'm indicating here by the strength of the color. So darker color indicating higher activity, and colors closer to white indicating less activity. So let's imagine that we have three different kinds of mushrooms in our minds. There may be different representations or different patterns of activity in the brain that reflect the encoding of those different objects. Now, the capacity to undergo a state change provides flexible and actuatable function to us as humans. So let's think about that state change. How does one state transition to another? And conceptually, I think about this as the movement from one vector to another vector. What features of neural systems constrain which of these transitions can happen and which transitions can't happen? And then also, what are the characteristics of a neural system that would allow some transitions to be relatively easy and other transitions to be relatively difficult? Let's think about this at a conceptual level, and then we can try to put it into a little bit of mathematics. So first, at a conceptual level, we're again thinking about the brain state as a vector of activity across either neurons at the cellular level, or for me, because I work predominantly at the large scale in humans and human neuroimaging, each of these elements would be a whole neural ensemble or a brain region. So beneath this activation state, there is a network of which populations of neurons are actually connected to one another. Right? So in here, I'm showing you that this particular pattern of activity has a network underneath of it where three ensembles are connected to one another. These three in the middle are less connected, and then the four on the end are more connected with one another. Now, we know that active neurons can spread activity to other neurons around them, particularly if they are connected. So having an active population is easy if the neurons in that population are connected. All right, well, that helps us to understand a little bit about which kinds of states might be easy to attain given an underlying network architecture. And I want to draw your attention first to the top left of this slide, where I have a pattern of activity, which is the first five ensembles being active and the second five ensembles being less active. And underneath of it, there's a network that has two modules, one on the left and one on the right. So this activity state is very easy for this network because all you have to do is activate this set. They share that activity among each other, and you have to inhibit the set on the right, and they remain inhibited because nobody is sharing activity with one another. So that's relatively straightforward. Let's now think about some state network pairs that are difficult or that provide some challenges. So at the bottom on the left-hand side, I've kept the same state active and inactive, but now the network underneath doesn't have that clear modular delineation. It has a lot of interdigitation between the two populations. For this network that has many connections between the populations, it's very hard to turn one set on and turn the other set off, because as soon as you turn this one on, it feeds all of that activity into the off section, and all of those populations become active. So this state is difficult for this network to reach. On the right-hand side at the bottom, I have changed the activity pattern that I'm asking the network to perform. So now I have an active population, an inactive population, active, inactive, etc. And the network that I want to perform that activation profile is the one at the bottom, which is highly modular. Now, this is tricky because, again, as soon as I activate one of these ensembles, it will spread that activity to the other populations that it is close to. And so having one active and the other inactive is difficult. Okay, so I've been using the terms easy and difficult to describe a single state, but I think we can also use them to describe transitions between states. So what's an easy transition and what's a hard transition? At the top, again, this is just conceptually, we may have an activity profile which is active and then inactive on top of a modular organization that allows that separation of activity to be easy. And if we want to change to having the first population inactive and the second population active, that's pretty straightforward too, because all I need to do is send one activation signal to the right-hand side and send an inhibiting signal to the left-hand side. What kinds of transitions are really difficult? Well, at the bottom, I'm showing you an example of a difficult transition. Here, the activity pattern is already hard to maintain. It's this active-inactive sequence. But then also, if I want to take that and switch it to anything that was active over here is now inactive, and anything that was inactive here is now active, that means that I have to have control signals at every single point in this network to deactivate and activate appropriately. I can't use anything about the network structure to help me attain that state transition easily. So I've been using the terms excite or inhibit, but now I want to sort of show you pictorially what I'm imagining in this conceptual framework. So if I wanted to have this transition at the top from the active set to the inactive set, all I need to do, as I said, was inhibit the first ensemble or module and activate the second one. But the reason that the transition at the bottom is very difficult is that I need to have an inhibitory signal or an excitatory signal at every single population. So the number and extent and strength and type of the control signals that are required for a state transition is a reflection of the ease versus difficulty of that state transition given the underlying network. Now, with that conception in your mind, I want to formalize it with a little bit of math and show you how we can use that to try to understand the brain. So there's a field of systems engineering, linear systems, and the sort of adjacent field of network control theory, which I think is really helpful here in operationalizing those concepts with a little bit of mathematics. So here on the left-hand side, what we have is a state of the brain at time t+1. So this state x right here is that vector of activations that I've been discussing, reflecting a state of the brain. And what we can do in a very simplified theory first is to just say that that state is equal to the state of the brain at the previous moment, so x of t, and then times a connectivity matrix. And this connectivity matrix we take to be the pattern of white matter connections between different areas of the human brain that we typically estimate using diffusion weighted imaging, and then following that imaging up with tractography. So what this multiplication indicates is that the activation of a single region can flow through a white matter tract to activate another region. Now, the last section of this equation indicates the kind of control signals, inhibitory or excitatory, that may be coming online throughout a particular state transition. Here, B sub kappa is a matrix with ones along the diagonal where the node to indicate which region is actually being activated. And then U sub kappa is the control energy that's injected into kappa regions at time t. Now, that is a way of assessing sort of the dynamics of the system, and it allows us to ask questions about the ease versus difficulty of a state transition. So just using that model of network dynamics, we can now define a cost function which penalizes the control energy required to get to a particular state, and that's what you're seeing over here on the right-hand side. So we're going to try to minimize this entire function, and that last term is the energy that's required for the state transition. We also want to minimize the distance of the current state x from the target state. So pictorially, I want you to focus on the left-hand side for the intuition. So pictorially, if we have the brain at a state x naught, and normally it would sort of flow along this gray line to some target state xt, that's fine, but maybe there's actually some input that is coming into the system that would push it in a different direction. And that is along this colored trajectory to a different x of t. And the question that network control theory can pose and answer is, given a network system, which of these state transitions is more difficult, requires more energy, and how would it actually happen? So bringing us back to the conception that we started with at the beginning, if we have a state of the brain that reflects a mushroom, then there's some energy that is required to transition to a new state reflective of a different mushroom. And then there's some additional energy required to move to another state. But all of these energy requirements are dependent on the underlying network architecture of the neural system, which constrains which transitions are easy, so small minimum energy, and which transitions are hard, so large minimum energy. An additional question that you can ask quantitatively with these approaches and this theoretical construct is, how can we maintain a brain state, and how much energy is associated with maintaining that state? So how can we persist in one particular state? Persisting in a state requires energy too, particularly if that state is not on an energy minimum of the accessible landscape. So with that, construct in mind, I'd like to ask the question of how we can use this idea of state transitions and the formal assessment of energy requirements to understand the progression of cognitive control in normative development as kids grow from a young age into adulthood, and also in the psychosis spectrum. And I'll be describing two studies, one that is now published and one that's a preprint available on medRxiv. So I want to ask the question of what sorts of transitions are important for the brain. So the brain may be sitting in one particular state, which is here on the left-hand side, and we want to ask, how many different states does the brain need to get to, and what sorts of transitions are important? Well, I think the answer is lots of transitions are important for a brain. Human brains are very complex and need to enact many different transitions and control many different transitions. So I think we need to be able to study not just one transition, but also many. So how do we do that? Well, there's a really nice approach in the field of network control theory that allows us to probe how you can take a neural system and push it either nearby on the local energy landscape of state transitions or far away on that energy landscape. The first one, the ability to push to nearby transitions, is actually something that you can derive from what's called the controllability Gramian. So this equation at the top is just the simple linear dynamical equation that I showed you earlier. And from that, you can write down the controllability Gramian, which, as you can see, is a function of the connectivity, the connectome A, and then B, which it tells you which regions are actually having energy injected into them. The average controllability is the trace of the inverse of the Gramian. So for anybody who likes the mathematics, that's the definition. But for those of you who want the conception more, it is the ability to push the brain into many local, easily reachable states on the energy landscape from wherever you're currently sitting. And now the ability to push to a distant state far away on the energy landscape is referred to as modal controllability. And that you can actually derive directly from the adjacency matrix, its eigenvectors and eigenvalues. But I want to show you what that looks like on the brain, which parts of the brain, when activated, would be able to push the brain system into lots of local, easily reachable states versus many distant states. So on the left-hand side, what you're seeing are the controllability profile of regions in what's referred to commonly as the default mode system. So this is a set of areas colored in blue on the brain at the top, which is active particularly at rest in a healthy human brain. And what you can see is that the levels of average controllability there are quite high, whereas the levels of modal controllability are quite low. And that indicates to us that regions that are active at rest in a healthy human brain show white matter connectivity patterns that are predicted to effectively drive the brain into many of the states nearby that it needs to reach. And I want to contrast that fact with what happens in the frontal parietal system, so a set of regions in frontal cortex and parietal cortex that are commonly activated in tasks that require cognitive control and executive function. And what we see in those areas is that there's a high level of modal controllability and much less average controllability. And what that means is that these areas have white matter connectivity patterns that make them structurally predisposed to be able to push the brain into very difficult to reach states, consistent with the transient control processes that these regions actually engage in for us as humans. Now we can ask the question of, if network controllability is useful for understanding cognitive control, is there where should we see it change? And a very obvious place that we should see it change is during development, when cognitive control and executive function are changing rapidly and extensively. So we actually studied a set of individuals between the ages of 8 and 22, which were part of the Philadelphia Neurodevelopmental Cohort, which was a large study funded by the NIH, led by Rick Helger and colleagues at the University of Pennsylvania. And every data point here is one of the youth in that study. And what we did is that we took their diffusion imaging data, we performed tractography, and we estimated the amount of modal controllability and average controllability in each one. What we see is that average and modal controllability are correlated with one another. So brains that have the capacity, theoretically, to push into local states are also the brains that have the capacity to push into distant states as well. So these two kinds of control strategies do not appear to be in a trading off with one another. They seem to be changing in concert throughout development. And the second thing that I want to mention is that both types of controllability are, the values are much greater than expected in null models. And we use two different null models that preserve either the node strength, which is the average weight of connections coming out of a region, or the node degree, which is just the number of connections that are coming out of a region. And those null models are shown in these yellow and peach ellipses, whereas the data points are from the true imaging data. We also then find that network controllability in this same group changes with age. So this is the exact same data, but replotted so that you can see the age of the individual. In dark blue are the youngest individuals, 8 years old, and in bright yellow are the oldest individuals, so close to 22 years old. And what you can see is that the younger youth tend to have low values of modal and average controllability, and the older youth tend to have much higher levels of modal and average controllability. So that indicates to us that these kinds of control strategies are increasing with development over the same time period in which cognitive control is changing appreciably. That happens at the cost of a decrease in global synchronizability of the system, which we define as the inverse variance of the eigenvalues of the Laplacian, but conceptually is just a reflection of how easy it is to have all of the units in the brain active and synchronized in their activity together at once. You might imagine that global synchronizability may not be particularly helpful, actually, for a brain, that more complex dynamics are needed to allow the repertoire of behavior that we engage in as adults. Now, an interesting and important question is this change in controllability something that explains the developmental patterns? Is it sufficient to explain the developmental patterns of changing white matter? And to answer that question, what we did is that we took each brain network that was derived from the diffusion imaging, and we rewired it in silico to push it towards higher levels of modal and average controllability. And we asked, do we end up with a white matter network that's consistent with what we see in the 22-year-olds? If we can rewire an 8-year-old brain along these two variables and arrive close to what looks like a 22-year-old brain, then that would suggest that these variables are sufficient to explain the developmental pattern. It doesn't mean that they're the only explanation, but it does mean that they are a sufficient explanation. So that's what you're seeing on the bottom here is the result of this multi-objective evolutionary algorithm, where we can take a brain and rewire it in silico to enhance these two features. And what we see is that the pink and yellow envelopes contain all of the data points, which are colored in purple, meaning that these variables are sufficient to explain the developmental patterns. There isn't some strange third variable that may be driving these effects. So that raises the question of, all right, so network control is changing over the same time scale that cognitive control is changing, but we don't really understand yet what it is about a white matter network that would allow it to exert more or less control. So let's think about that a little bit. In this paper that Jason Kim led now a little over a year ago, what we found is that for the whole brain, there are some patterns of connectivity that make control more difficult and make state transitions more difficult. And those are patterns of connectivity where a single region of the brain is connected to another region, but not to anywhere else, or where brain regions have similar weights on their edges, meaning the same strength of white matter connectivity between two regions. And I want to contrast that sort of difficult picture up at the top here with a pattern of connectivity that makes control easier. And that's at the bottom here where I write easy. So this is where regions have many connections to other areas, and those connections all have different weights. So we find that dissimilarly distributed weights on these connections between regions decreases the energy required for control. So that complexity that we observe in the older youth is something that helps make control easier. Now that's what happens at the whole brain level, but for a single brain region, there's a similar story, which is that regions that have very strong connectivity to other regions tend to have high average controllability. So I want you to notice that I'm going to use this term strength in the next two slides, and strength is the average weight of connectivity coming out of a region. And then on the right-hand side, what I'm showing you is average controllability takes into account that local strength, but it also takes into account more complex network organization farther away from the immediate brain region of interest. So I want to ask the question of whether strength or average controllability are important and where they are important in the brain to understand control. And what I'm showing you up here at the top is a gradient. This is the principal gradient from unimodal areas in the dark bluish purple to transmodal areas in the bright yellow. And what you can see is that the correlation between a region's strength and its average controllability is decreasing as you go from unimodal regions to transmodal regions. What that means is that in unimodal cortex, let's say in visual cortex and motor cortex, how strongly connected those regions are locally is a good predictor of their average controllability as well. But that's in contrast to regions in transmodal cortex, in frontal and parietal areas where the connectivity surrounding the local area is actually also very important and very complex and drives the control properties in unexpected ways. And so that raises the question of, well, these are also areas that are developing a lot over that period, these transmodal areas, and they are also areas that are important for cognitive control and whose activity is altered in many psychiatric conditions. So we wanted to ask whether average controllability, because it seems to be more sensitive to the complexity of connectivity in these transmodal areas, is important for understanding the psychosis spectrum. And this is work that was led by Lyndon Parks in the lab, and again is available as a preprint on medRxiv if you want to dig into the details. But what we wanted to do was to ask whether we could predict psychosis positive symptoms from control profiles. And we used a prediction model where X was used to predict Y. So in this case, we're going to try to predict psychosis positive symptoms, and we're going to try to predict them from either average controllability, which is sensitive to these complexities of connectivity in transmodal cortex, or from a simple strength measurement. We're going to control for age, for sex, for brain volume, and in scanner motion. And we'll use 100 repeats of 10-fold cross-validation, each repeat using a different random split of the data to sort of ensure reliability of the results. And what we find is that there's a robust prediction of psychosis positive symptoms from average controllability, and significantly more so in transmodal areas. So what I'm showing you along the y-axis here is the root mean squared error, and that is the prediction is better when the RMSE is small. So the fact that we have low values of error in transmodal cortex indicates that transmodal average controllability is predictive of, and significantly predictive of, psychosis positive symptoms. We can contrast that with strength, which is just a simple measure of local connectivity, and we can see that strength is really not as predictive of psychosis positive symptoms, and it also is not very sensitive to variations from along the principal gradient from unimodal areas to transmodal areas. So this study indicates that network controllability, particularly in transmodal cortex, predicts psychosis spectrum symptoms in a group of individuals who is includes many of the Philadelphia neurodevelopmental cohort individuals and some follow-up people acquired through similar means, again, led by Raquel Greer. And all in that same age range, 8 to 22. Now, as we move into the very last section of this work, I wanted to use the theory not just to probe normative development and also the psychosis spectrum in normative development, but to probe executive function and dysfunction in adulthood. And this work I will be covering is led by Zhai Zhu. It was published in eLife this year, and the senior author on this paper is my colleague Ted Satterthwaite, who's in the Department of Psychiatry at University of Pennsylvania. And I'm also going to cover some work by Urs Braun, who is now an assistant professor at the Central Institute of Mental Health in Mannheim, Germany. And this was work whose senior author is Heike Toast, and also another senior author is Andreas Meyer-Lindenberg. So these two are collaborative efforts. So what transitions are important, not just for the brain, which is how I opened the last section, but for executive function specifically? I really want to ask the question of how the brain gets from where it currently stands to activation of the cognitive control circuitry. So I'm just going to focus in on one particular state. And to focus in on that one particular state transition, I'm not going to use average and modal controllability anymore, because those techniques allow you to ask about many local transitions or many distant transitions, but they are not sensitive to the precise initial and target states. Instead, what I'm going to use is this control trajectory approach, which allows me to specify the current state of the brain and the target state of the brain, and ask the question of how much energy needs to be injected in order to push the brain from here to here. So let's imagine, for example, that we have a brain at baseline without any particular activation profile. And what we want to do is to activate the frontal parietal cognitive control system. So here's a picture of what that looks like. We would start at baseline in the initial state, inject energy into the system to move it to the frontal parietal activation. And we will do this in the context of, again, the Philadelphia neurodevelopmental cohort. And in this case, we've expanded from 822 individuals to 946 individuals, again, between the ages of 8 and 22 years. This setup allows us to isolate the effect of activating the frontal parietal system, and it uses single subject white matter connectivity network, so that we can say, while this transition is the same transition for everyone, its difficulty may be different for each person depending on how their white matter is structured. So what we find is that the control energy for one of these state transitions decreases as a function of age. So consistent with the fact that the engagement of control circuitry happens very frequently and more easily in older youth. So what you see here is control energy. You see it decreasing as a function of age for this transition to the frontal parietal activation. This evidence supports, but does not prove, that the white matter network develops to ease the transition to executive function, thus facilitating the frequent use of this cognitive process in older youth. Now we want to ask the question of not just how much control energy is changing with age, but if you remove the effect of age, can it explain individual differences in cognitive control performance? And to answer this question, we used a neurocognitive battery, the Penn computerized neurocognitive battery, and a factor analysis applied to it to extract an executive performance factor. And that's what you're seeing along the x-axis here. So executive performance, a z-score of executive performance against the control energy along the y-axis. Again, you see a negative relationship. And what this means is that the individuals who perform quite well on the tasks that require executive performance are those who also require less control energy to activate the frontal parietal system given their white matter connectivity. We also found that the bilateral middle cingulate mediates the relationship between age and executive function. So here you see a formal mediation model between age and executive function, where the control energy in the middle cingulate is a significant mediator of that relationship. So what that suggests to us is that there's additional evidence that the framework of network control and the amount of energy that we can estimate to be necessary for a transition is consistent with behavior, and in fact helps to explain this relationship between age and executive function. Now, I had mentioned earlier in the very first part of the talk that the approach doesn't only allow us to assess transitions between two states, but also how much one state can be held in the mind or in the brain for a long period of time. So the persistence of a state. And to understand sort of the requirements to change states, but also to hold a state steady, Urs Braun from the Central Institute of Mental Health in Mannheim, Germany, decided to evaluate the common working memory task that is often used to understand executive function in healthy humans, but also in humans that have psychiatric conditions. And what you see here is the idea that in a common working memory task, we may have a block of the task that is a zero back task, where individuals are simply responding to the visual stimulus that's in front of them. And then that may transition to another block, which is a two back task, where individuals need to remember the stimulus that was two back in a series. What he did was that he asked how much energy is required to move from the zero back state. So the pattern of activation in the zero back state, assessed using a simple general linear model commonly used to understand brain state activation in fMRI data, to the two back state. And what you can see here is the predicted control, the estimated control energy along the y-axis, if we are moving from the zero back state to the two back state, or if we are moving from the two back state back down to the zero back state. And what's really interesting is that the transition to the more cognitively demanding state, the two back, requires more control energy than the transition down to the zero back state, so the simpler state that requires less sort of cognitive load. And that's consistent with how we think about these tasks. So control energy appears to be tracking in an associative way, the cognitive load of the task. More control energy being required to hit the high working memory load state, and then less control energy required to fall back down into the zero state. We can also ask whether the zero back state or the two back state is easier to hold online for a long period of time, given the white matter underneath. And so we can estimate this stability measure, which is how the inverse of how much energy is required to hold the state. What we find is that the stability of the 0 back state is significantly higher than the stability of the 2 back state. And that means that there is much less energy required to hold 0 back and much more energy required to hold or persist in the 2 back working memory state. Interestingly, we also found that, as you can see, there's a lot of individual differences here. Every data point here is a different person. And we found that those individual differences are predictive of performance. So greater stability of the 2 back state was associated with a higher task accuracy across individuals. Lastly, Urs wanted to ask the question of what is the underlying neural mechanism for some of these large-scale systems level estimates. And he hypothesized that the ease or difficulty of these transitions, particularly in the working memory task, would be related to dopaminergic signaling. So he used a randomized placebo-controlled double-blind pharma fMRI study, particularly using amisulpride, a selective D2 antagonist, and he found that that drug increases the control energy for both transitions versus placebo. And to show you that, I want you to focus on the left-hand side of this slide where we have the control energy. And then in the darker color is placebo, in the lighter color is amisulpride. In the first section of the figure, you can see the transition from 0 back to 2 back. And we find greater control energy required in 2 back in the amisulpride condition. And then you can see in the 2 back to 0 back condition, again, we find that more control energy is required when individuals are on the amisulpride than when they were given the placebo. Then lastly, he thought, we know that schizophrenia patients have marked dopaminergic abnormalities, suggesting that perhaps this control energy is going to be altered there. They also have deficits in executive function, and they also have alterations in structural connectivity, so white matter tract organization. We find, so he decided to consider individuals who have schizophrenia and ask to what degree their control energy may differ from healthy controls in these same state transitions. And he finds an effect that's similar to similar in direction to that of the amisulpride, particularly in the 0 back to 2 back transition. So on the right-hand side of this slide, you can see the healthy controls in the lighter blue and patients with schizophrenia in the darker blue. And you can see in this transition from 0 back to 2 back, so towards the higher working memory load or requirements, greater demands on the executive system, the control energy required is much higher for people with schizophrenia than for controls. We did not see a difference in the transition from 2 back down to the 0 back condition. So that provides a new lens, I think, into the relevance of the network control approach for understanding executive function and its dysfunction. So I just wanted to sort of broaden back out and say that I could have titled this talk Structural Constraints on and Support for Control, because we really focused on how underlying white matter connectivity may constrain the ease or difficulty of state transitions, and that's what we talked about in the first section of the talk. Then we described how that white matter connectivity can explain the alterations in cognitive control during normative development. And we also discussed some evidence for its relevance in understanding the psychosis spectrum. And then lastly, we moved into using that theory, this network control theory based on underlying white matter, to probe executive function and dysfunction. But the take home that I really want you to have in your mind as we leave and move into the question and answer session is this very big picture conceptual framework that the brain has states, it moves between different states, and the energy that is required for those state transitions can depend markedly on the underlying architecture of connections between neural units. So very precisely, the network architecture of neural systems constrains which transitions are easy, so have a small change in energy, versus which transitions are hard, having a large change in energy with relevance, as we've seen over the talk, for development, for disease, and for the effects of drug. So with that, I'd like to acknowledge the members of the lab and obviously our funding bodies who have allowed us to do this work. I'd like to put a particular shout out to our collaborators, including, as I mentioned, Ted Satterthwaite, Raquel Gurr, and Urs Braun, Heike Toast, and Andreas Meyer-Lindenberg for some of those last studies. And with that, I'd love to stop and ask if you have any questions. Thank you so much for listening.
Thank you so much, Dr. Bassett. That was really a fascinating talk and just a remarkable integration across so many fields and presented so beautifully. So really thank you for that talk. I guess, I will get us started with one question. When in thinking about this concept of controllability, is your sense that this is applicable to specific domains within psychiatry, or does its application sort of depend on the brain networks that you're interrogating, and thus can have applicability across various psychiatric domains?
That's a really great question. I think it is the network control approach is definitely applicable across different diagnoses. I think it's particularly relevant when we think the alterations are distributed spatially throughout the brain and cannot be localized to a single area, which is sort of very common in psychiatry, but it may be less true for particular neurological conditions that have very focal pathologies.
The other thing I wanted to ask about is, and I got a little hung up on, is that in hearing your talk, it seems that there should be a relationship between controllability and one's capacity to learn and integrate new information. And yet the developmental course that you showed was that controllability seems to improve or increase with age. And that's somewhat contrary to the developmental process of learning, where children are less efficient at performance, but in terms of learning and integrating new information, at least my children are much more facile at it than I am. And so I'm curious to get your thoughts on that.
Yeah, that's a really good question. I think the two kinds of controllability that we study that do increase with age, so average and modal controllability, allow for a very complex dynamical repertoire of states that are accessible to the person. And so I think that that enhanced set of states allows for the complex behaviors that adult humans have. But you're absolutely right that learning and that sort of flexibility of learning is something that kids have in some ways better than adults do. My hypothesis is that there are classes of transitions that are not captured by those two that I discussed, that may be heightened in children and decrease into adulthood. And that's something that we haven't yet shown, but that I would hypothesize to explain this quandary that you raise, which I agree with.
Great. So this is a question from Gaurav Patel, and may also touch on the last point, which is, how much of these findings depend on the specific connectivity measures that you use? So you're looking at tractography as opposed to functional connectivity. And I guess commenting on why you chose tractography and how things might differ if you looked at different connectivity measures.
Yeah, that's a really great question. So the field comes from, it's actually called structural controllability in the network control theory literature, and it's commonly used to understand robotic systems and material systems and power grids and things where there really is a physical link between two power stations, and electricity can flow between them along the link. So it's been developed in the context where there's a specific structural link that pairwise connects one bit of the network to another bit of the network. I think that the challenge with functional connectivity is that often the statistics that we use to measure functional connectivity don't reflect pairwise relations that can't be explained by third parties or fourth parties or fifth parties. So for example, in fMRI data, often functional connectivity is assessed using a Pearson correlation coefficient, and that does not seek to isolate pairwise relations that are causal or even or more fixed. And so therefore the whole model that we write down doesn't really reflect the biology if the functional connection is a Pearson correlation. I think what the part that I think is possible is to use measurements of effective connectivity. So for example, structural equation modeling or dynamic causal modeling. So these are parts of the field or approaches that have been developed in the field to try to isolate pairwise relationships between regions that can't be explained by a third relation. And I think if you use those sorts of approaches, then the network control framework and the dynamical model I wrote down still has some biological relevance. In fact, we just had a paper that came out, I think it was yesterday, in the Proceedings of the National Academy of Sciences that uses effective connectivity and control. And it's in the context of seizure dynamics to say, during a seizure, does the pattern of effective connectivity change? And if so, how does that alter the controllability of the seizure itself, and with relevance for how to perform seizure suppression? So if you're curious about that sort of extension, I might point you there.
Wonderful. So another question also from Gaurav Patel is whether you've tried to get at causality of these models with anatomically targeted interventions such as TMS.
That's a really, really great question. We do have some work in TMS that we are in collaboration with Desmond Othis and Ted Satterthwaite. It's not a preprint yet, so I don't have the full story, but I think I will raise instead some work that Jenny Stizo in the lab led, which is actually using ECOG, so intracranial electrocorticography, and stimulation, so grid stimulation. And so what we can do is we can say, what's the pattern of activity before stimulation? Now you stimulate one sensor. What's the pattern of activity after stimulation? And is it consistent with our theory? And so that paper was published in Cell Reports last year, I think. So that's some nice initial evidence with a causal perturbation to assess the validity of the model.
The next question is from David Hellerstein, who says, thank you for the fascinating talk. I am interested in your thoughts on how network controllability may be affected by conditions such as major depression and how treatments might mitigate such disordered controllability. For instance, could psychedelic medications potentially improve network reorganization by disrupting disordered or overconnected networks and allow for more adaptive network reorganization?
Yes, I think this is a really fantastic question that we don't have answers to yet. So I guess if you want my opinion rather than scientific data, my opinion is that yes, when controllability is altered in a particular condition, then I think it could inform how we intervene. And then the question is, what sorts of interventions are relevant? Can you can, for example, use neurofeedback, or maybe cognitive behavioral therapy does a similar thing, which is to change the functional connectivity, or the effective connectivity, I should say, the effective connectivity between two regions, thereby altering the kind of control that's possible for that person. And so I'm really excited to sort of be looking into the future at opportunities to ask to what degree interventions informed by the control can help people, for example, in major depression.
Great. The next question is from John Mann, who writes, decision-making has a cost function in terms of energy, and some conditions are associated with lower energy availability that has consequences for decision-making. How can your model consider state fluctuations or individual variation in that available energy?
That is a really good question. I would say that one of the limitations of the model is that it doesn't explicitly have neurotransmitter levels included in it, and it also doesn't have any explicit assessment of differences in computation that various regions of the brain can perform. And so it is, I think, best used to understand how the connectivity constrains what can happen, but there are other constraints beyond connectivity. So I wouldn't say that as it currently stands, it would necessarily be able to address what John has just raised. I think there are really interesting extensions in the literature, in the engineering literature, I should say, that have not yet been brought over. And so I think there are nice possibilities for expanding the model, but they haven't been performed yet.
So there's several questions about how modifiable controllability might be. So I'm going to try to summarize some of those coming from Ralph Wharton, Rebecca Muley, and some others. So could you comment on individual differences in controllability, whether learned skills affect controllability, such as musical skill, reading, athleticism, any known effects of modifying controllability?
There is certainly significant variability in individual variability in controllability, and that changes over development, but we also have a paper that was published in Neuroimage. The first author is Eli Kornblath, shows that it is related to impulsivity. So in case you want to look there, but none of those studies assess interventions and training. So I don't yet have any data to provide there.
And also several questions about etiology. So William Tucker and others ask about whether any insights about genetic determinants, heritability, twin studies.
Yep, there's a really nice study from Sophia Franjo, I'm sure I'm pronouncing that wrong, that assesses heritability of controllability, and specifically average controllability, not modal controllability or any of the control energy requirements yet. So, but there is definitely some evidence for significant heritability of the average controllability, and that's her work.
Wonderful. Oh, sorry, wonderful, thank you so much. As I scan through some of the other questions, I was wondering, this may be difficult to do in a short period of time, but if possible, I think people would be really interested to hear a bit about the work that you've done on citation biases. So if possible to summarize, that would be wonderful.
Sure, absolutely, yes. And just to kind of raise again what you mentioned at the beginning, just for anybody who wasn't there, this is work that was led by Jordan Dworkin, who's now an assistant professor in the Department of Psychiatry at Columbia. So he's all with you all. So if you want to ask additional questions, you should feel free to do that. But yes, he was really interested in understanding the degree to which papers that are written by a person of a given gender are cited appropriately, or perhaps oversighted or undercited. And so what he did is that he studied reference lists from 60,000 papers in 5 journals, Nature Neuroscience, Neuron, Brain, Journal of Neuroscience, and Neuroimage. And he predicted the gender of the first author and the gender of the last author using probabilistic databases that assign a probability. If you have the name Danielle, you are more likely to be a woman than a man. And if you have the name Jonathan, you are more likely to be a man than you are to be a woman. And there are probabilities assigned to those names based on large databases from Social Security Administration database, and then also some international databases as well. So after predicting the gender of the first and last author, then you can ask the question of whether papers that are written by someone, one gender pair are cited in proportion to how many papers there are out there in the literature of that kind. So in other words, if there are 10,000 papers that have a woman as first and last author, and there are 6,000 papers overall that we studied, then you would expect a certain number of citations to be allotted to woman papers. So he showed that using that assessment, the papers that have a woman in either the first or the last author position are significantly undercited by about 30% by the field, and papers that have a man in the first and last author category are oversighted by about 25% in the field. He also did a more complex model that accounts for how old is the paper, because maybe we're citing older papers differently than newer papers. Is the paper a review article or an empirical article? What is the journal that it was published in? Was it in Neuroimage or was it in Nature Neuroscience? And even after accounting for these and the seniority of the authors, he found that the overall effects still hold. And the sort of concerning part of it is that the effects are increasing with time. So papers that are written by women are being cited less and less with time, and papers that are written by men are cited more and more over time. This is from 1995 to 2018. And the one last sentence I want to add is that these same trends are holding for authors of color. So we have a follow-up paper that's on bioRxiv if you're curious. And what we find is that papers that are written by an author of color in the first and last author position are significantly undercited. And most of that is being driven by the citation practices of white authors, so people like me. And it's also increasing with time. So it's suggesting to us that there's something that we as a community need to think about and address in the way that we acknowledge the work of others and the value that we place on the work of others.
Thank you so much for summarizing that. It's really such important and interesting work, and I suspect has implications or explains some of the issues around biases around funding at NIH, which is an ongoing struggle.
Yeah, unfortunately.
So there's a question from Diana Rodriguez is, thank you so much for the fascinating talk. I wonder if network controllability is determined also by the small worldness of structures within the network.
That is a really good question. They're somewhat distinct. So you can have a small world structure in a network that has binary edges and thus would not have particularly high controllability. So it goes significantly farther in describing the complexity of the weights on the edges and how those weights are distributed either local to a region or far away from a region. So there's not a clean relationship between those two concepts.
Okay, another question is from Jishun Wang. And what is the, I think you commented somewhat on this, but what is the main difference between your state model and dynamic causal modeling? I think you did comment on that in regards to the one being related, yours primarily being related to tractography versus dynamic causal modeling being from functional imaging. But if you have additional thoughts, please let us know.
Yeah, so the dynamic causal modeling does not explicitly take into account the structural connectivity, which ours does. But it also, I would say that the dynamic causal modeling is kind of trying to understand and identify the causal relationships between two regions. It doesn't necessarily, which is distinct from but complementary to the questions that we're posing, which relate to how you can push a brain from one state to another. So we are asking more about how you guide state transitions or how that guidance happens intrinsically in a healthy human, whereas dynamic causal modeling is seeking to infer a causal relationship between two or more areas. The goals are quite different, but yes, you could combine them by using dynamic causal modeling to infer effective connectivity and then use that effective connectivity to understand state transitions. You could sort of use them in parallel, or in series, but although I haven't seen anybody do that yet.
So thank you so much. I think that wraps it up for our questions. And I again wanted to just comment on how exceptional your work is in your capacity to integrate across fields, bringing so many disciplines together and to be able to ask such impactful questions for psychiatry. So thank you so much for your time and sharing your thoughts with us.
Thanks so much for having me.